Module 4, Forensic Toxicology, Unveiling Secrets. Forensic toxicologists, the unsung heroes of the crime lab, play a crucial role akin to that of a skilled detective. Their primary task is to detect and identify chemicals in the body that could have contributed to a crime or a sudden, unexplained death. Imagine a scene where a toxicologist, like a modern-day Sherlock Holmes, examines bodily fluids and tissues, searching for the tiniest clues that chemicals leave behind. These professionals don't just identify substances, they also interpret the findings. Was the victim poisoned? Was the driver under the influence? They answer these questions and more, providing critical information in legal and criminal contexts. Now let's raise a glass, figuratively, of course, to understanding how alcohol travels through the body. When you sip your favorite drink, alcohol doesn't just magically make you feel tipsy. It's absorbed primarily through the small intestine, then hitchhikes via the bloodstream to party around your body. It affects the brain, lowered inhibitions, but it doesn't get to crash there indefinitely. The liver acts like a responsible friend, metabolizing, breaking down the alcohol mostly into water and carbon dioxide. What the liver can't handle, the body excretes through urine and breath. Yes, you breathe out alcohol too, not just the morning after. Measuring blood alcohol, determining if someone is under the influence, isn't as simple as asking, "Have you been drinking?" Forensic toxicologists use various methods to measure blood alcohol concentration, BAC. Breathalyzers are like the party bouncers. They're the first line of defense in checking if someone's over the limit. But the real deal is blood testing. It's more accurate and gives a clear picture of how much alcohol is cruising in someone's bloodstream. These tests look for ethanol, the type of alcohol in beverages, and can indicate whether someone's BAC was over the legal limit at the time of an incident. You have the right to remain silent, but not to refuse a test. Implied consent and legal intricacies. Navigating the legal aspects of blood alcohol testing is like walking through a constitutional maze. The implied consent law, as recommended by the National Highway Traffic Safety Administration, is a fascinating solution to this puzzle. Essentially, when you get your driver's license, you're implicitly agreeing to undergo blood alcohol testing if suspected of drunk driving. Refuse the test, and you might face penalties like license suspension. This law cleverly balances individual rights with public safety, addressing the constitutional concerns about forced testing. Forensic toxicologists are not just alcohol detectives, they're also adept at uncovering drugs and poisons. Their toolkit is like a magician's box, full of techniques to isolate and identify various substances. They use screening tests to get a broad idea of what might be present. Think of it like sniffing a stew to guess the ingredients. Then they turn to confirmation tests, which are like following a detailed recipe to pinpoint exactly what's in that stew. They use sophisticated instruments like gas chromatography and mass spectrometry, big words for even bigger roles in identifying the tiniest amounts of substances. Hair and fiber forensics. Hair, often seen as a mere accessory to personal style, hides an intricate story beneath its surface. Each strand of hair experiences a cyclical saga of growth, rest, and eventual loss. This tripartite drama unfolds in 3 acts: anagen, the phase of active growth; catagen, a transitional phase; and telogen, the resting phase. It's during the anagen phase that hair reveals the most about an individual's lifestyle and health, much like a diary, chronicling the effects of drugs, toxins, and nutritional habits. But what makes hair so telling? The answer lies in its architecture. Human hair is a complex assembly of the cuticle, cortex, and medulla, each layer adding its own chapter to the hair's story. The pattern of pigmentation, the medullary index, and the type of cuticle vary dramatically between human and animal hair, turning each strand into a unique identifier of its owner. When hair pirouettes onto the forensic stage, it demands a meticulous examination routine. The investigator embarks on a microscopic waltz, observing the hair's length, color, and diameter. Chemical tests may follow, sniffing out any toxins or drugs that have danced their way into the hair's composition. This evidence, while not a fingerprint, is a powerful chorus in the symphony of class evidence, adding depth and context to the investigation. In the meticulous art of evidence collection, hairs are treated like fragile relics. Whether plucked by forceps or lifted by tape, Each strand is a potential goldmine of information. The collection process is akin to a careful excavation, ensuring that the evidence remains pristine and unaltered, ready to tell its tale under the microscope. Fibers, the unsung heroes in forensic science, come in a diverse wardrobe of types. Some fibers, like cotton and wool, are the natural confidants of plants and animals. Others, like nylon and polyester, are born from the crucibles of human innovation. Each type of fiber, whether natural or synthetic, carries its own signature properties, a testament to its origin and journey. In the theater of forensic comparison, fibers are scrutinized for their color, diameter, and the peculiarities of their surface texture. It's a bit like comparing two actors auditioning for the same role. Some properties make them perfect for the part, while others set them distinctly apart. These properties, whether the fiber's refractive index or its dye composition, become critical in unraveling the mystery of their journey from a crime scene to their source. Fiber collection is a choreography of precision. Investigators sweep the crime scene with vacuums, dab with tape, or gently pick with forceps, ensuring that each fiber is collected without contamination. Once gathered, these fibers are preserved as if they are delicate artifacts, ensuring their stories remain untainted and ready for the forensic stage. Forensic analysis and preservation. Examining a painting in a forensic context is akin to unraveling a historical puzzle. Each brushstroke, pigment, and canvas thread holds clues to its authenticity, origin, and condition. In the laboratory, paintings undergo meticulous examination. Techniques such as infrared reflectography, X-radiography, and UV fluorescence help in uncovering underdrawings, previous restorations, and hidden details invisible to the naked eye. Preservation of these artworks is a delicate dance between science and art. The primary aim is to prevent deterioration and maintain the painting's integrity. This involves controlling environmental factors such as humidity, temperature, and light exposure. Often, conservators use reversible materials for restoration, ensuring future generations can retreat the artwork with advanced techniques. Shattered clues, forensic glass evidence. Glass evidence, often found at crime scenes involving break-ins or accidents, can provide invaluable information. The collection and preservation of such evidence are critical. When collecting glass fragments, forensic experts meticulously document and photograph the scene, ensuring the spatial relationships and conditions are recorded. They wear gloves to prevent contamination and use forceps to pick up larger pieces. Smaller fragments are carefully swept into a container. Preservation involves storing these fragments in rigid containers to prevent further breakage. This careful handling is crucial as the glass's physical properties, such as fracture patterns and density, can reveal the direction and force of impact. One fascinating aspect of glass evidence is fracture analysis. Glass tends to break first on the side opposite the strike, creating radial fractures that radiate outwards. This can indicate the direction of the force applied. Additionally, forensic experts often compare the refractive index of glass fragments from the crime scene with those of suspected sources. This analysis can link a suspect to the scene or exclude them from consideration. The ground beneath, forensic soil analysis. Forensic soil analysis is like piecing together a story from the very ground we walk on. Soil, as unassuming as it might seem, can be a treasure trove of evidence. It contains minerals, organic matter, and sometimes even microorganisms unique to a location. The collection of soil evidence requires precision. Forensic scientists collect samples from various depths and locations at the crime scene, ensuring they represent the area comprehensively. These samples are then compared with soil from a suspect or their belongings. In the lab, soil analysis involves several steps. Scientists examine the soil's color, texture, and composition. Microscopic analysis can reveal the types of minerals present. For instance, quartz is a common mineral found in sand across different locations. Advanced techniques like gas chromatography and mass spectrometry can further identify organic compounds, offering clues about the soil's origin. Pollen analysis, or palynology, is another intriguing facet of soil forensics. Pollen grains, unique to specific plants and regions, can create a pollen fingerprint of a geographical area at a particular time of year. This can link a suspect or object to a specific location. The plant world also offers a myriad of clues in forensic investigations. Angiosperms, or flowering plants, are particularly diverse, including species like corn, oaks, and maples. With over 300,000 known species, they provide a vast pool of evidence. Two key parts of the flower often examined are the stamen and pistil. The stamen, consisting of the anther and filament, is the male reproductive part, while the pistil, the female part, produces eggs. Understanding these components is crucial in forensic botany, as they can link evidence to specific types of plants found at a crime scene or on a suspect. This type of analysis requires a keen eye and a deep understanding of plant biology, biochemistry, and forensic science. Imagine a lock-and-key mechanism at the molecular level. That's essentially what antigen-antibody interactions are. In the fascinating world of forensic science, this concept plays a crucial role in species identification and drug identification. Antigens, which could be proteins, polysaccharides, or nucleic acids, act like the lock, while antibodies, produced by the immune system, are the key. These interactions are highly specific. An antibody formed against a specific antigen will only bind to that antigen, much like a key opening a specific lock. When it comes to species identification, this specificity becomes a powerful tool. For instance, if a blood sample at a crime scene contains proteins unique to a certain species, antibodies specific to those proteins can be used to determine the species of origin. Similarly, in drug identification, antibodies can be designed to bind to specific drug molecules, thereby identifying the presence of those drugs in biological samples. Characterizing blood stains in forensic analysis. Blood, a vital piece of evidence at many crime scenes, contains a wealth of information. But first, forensic experts must confirm that a stain is indeed blood. Several tests exist for this purpose. The Kastle-Meyer test, for instance, uses the chemical phenolphthalein, which reacts with the hemoglobin in blood to produce a pink color. Luminol tests, on the other hand, rely on the luminescence produced when luminol reacts with iron in the hemoglobin. While these tests indicate the presence of blood, they cannot distinguish between animal and human blood, a task that requires further analysis, such as antigen-antibody interactions. In cases of sexual assault, seminal stains are crucial evidence. To characterize these stains, forensic labs use a series of tests. The acid phosphatase test, which detects an enzyme abundant in seminal fluid, is a common starting point. Following a positive result, more specific tests like the P30 test, which identifies a protein unique to seminal plasma, are employed. Microscopic examination can also reveal the presence of spermatozoa, although this is not always conclusive, since some individuals may have low or absent sperm counts. In rape investigations, the collection and analysis of physical evidence are paramount. This involves gathering clothing, bedding, or any other materials that may contain biological evidence. Medical examinations can yield vital evidence, such as semen, saliva, or blood, which are then analyzed using techniques like DNA profiling. The sensitivity and specificity of DNA analysis make it a powerful tool in identifying the perpetrator, especially when combined with other pieces of evidence. The structure of DNA, with its double helix and sequence of nucleotides, encodes the blueprint of life. Each sequence of 3 nucleotides, called a codon, corresponds to a specific amino acid, the building blocks of proteins. The process of translating the DNA code into a protein involves transcribing DNA into RNA, and then translating RNA into a sequence of amino acids. This intricate process is fundamental to life and forms the basis for various genetic analyses in forensic science. Polymerase chain reaction, PCR, is like a genetic photocopier, amplifying tiny amounts of DNA to quantities sufficient for analysis. Short tandem repeat, STR analysis, then comes into play, focusing on regions of DNA where certain sequences are repeated. Since these patterns vary greatly among individuals, STR analysis can provide a genetic fingerprint unique to each person. Gel electrophoresis, on the other hand, separates DNA fragments based on size, providing a visual representation of STR analysis. These techniques revolutionize forensic investigations, allowing for precise identification even from minute samples. Fire and explosion chemistry. Let's start with fire, nature's own drama of chemical reactions. Fire, at its heart, is a love story between fuel, oxygen, and heat. When they meet, it's chemistry. The initiation of a fire involves an exothermic reaction, fancy science talk for it releases heat. Molecules of fuel break down, oxygen swoops in, and voila, combustion occurs, producing heat, light, and new chemicals like carbon dioxide and water vapor. But what keeps this fiery tango going? It's all about sustaining the reaction. As long as fuel, oxygen, and heat are in abundance, the fire continues its dance. Take one element away, and the fire bows out. Picture yourself as a detective at a charred scene. Your mission? Find where the fire started, the point of origin. It's like reverse engineering the story of the fire. Look for the area with the most damage. It often whispers the tale of the fire's beginning. Burn patterns, melted materials, and the direction of smoke and soot can lead you there. Now about accelerants. These are the plot twists in fire stories, substances that were invited to make the fire bigger and hotter faster. They're like fuel on steroids. Telltale signs? Deep charring, melted metals, and the aha evidence, irregular burn patterns. Collecting evidence at an arson scene is like gathering pieces of a puzzle. It requires a keen eye and a gentle hand. Wear your gloves and think like a scientist. Collect fire debris, especially from the origin area. Look for containers, wick materials, or any out-of-place item. These are your clues. Remember, the scene is your library, and each charred piece is a book with a story. Package the evidence properly to prevent contamination or loss of volatile substances. And yes, that pungent smell could be your lead to accelerants. Back in the lab, the focus shifts to detecting hydrocarbon residues, the remnants of accelerants. How? Through a symphony of scientific techniques. Gas chromatography mass spectrometry is the star performer here. It separates, identifies, and quantifies chemical substances with precision. A sample from the fire scene is vaporized and sent on a journey through a column. Different substances travel at different speeds and are detected at the end. It's like a race where each runner molecule has a distinct running style, chemical property, making it easy to identify them. Explosions, the louder cousins of fire, are dramatic tales of rapid energy release. Chemically, it's a rapid oxidation reaction, oxygen mingling with other substances in a hurry, releasing gases, heat, and sound. Unlike the sustained dance of fire, an explosion is a sprint, a rapid release of energy in a powerful burst. Post-explosion, the scene is like a scattered jigsaw puzzle. Collecting evidence here is a delicate task. Look for fragments, residues, and anything that seems out of place. The pattern of debris can tell you about the strength and location of the explosion. Back in the lab, the fragments tell their tales. Chemical analysis, much like an arson investigation, plays a pivotal role. Identifying the type of explosive used often involves detecting specific chemical residues. Techniques like ion chromatography and infrared spectroscopy come in handy, revealing the explosive's chemical fingerprint.